Hi. So we have learnt various host pathogen interaction, and in that series we have learnt about the host virus interaction, host bacteria infection. We have learnt about the host fungal infection, and now we are reaching towards end, and we will learn about the host parasite interaction. Okay. So there are variety of parasites, okay, and these parasites are causing quite serious diseases in human as well as in some animals as well. So we are going to discuss about these parasites. So in this session, first I will introduce about parasites, and then we will take one parasitic disease which you are very well aware, that is malaria. We will take this malaria in great detail. So after that we will discuss also about African sleeping sickness, which is also a quite serious disease, and we will finish this course. Okay. So let us begin with the parasites. Okay. So there are two major kinds of human parasites. Okay. So one parasite we categorized as a protozoa, okay, and probably you are aware that protozoa is a unicellular eukaryotes, okay. They have a proper nucleus, okay. That's why we call it as a eukaryotes, okay. They replicate within the host, these parasitic protozoan, okay. They replicate within the host. There are also various free living protozoa, right, like paramecium, probably you might studied in class 10th or 12th standard, or there are amoeba, okay. But these, the parasite which we are going to discuss, or I am going to discuss, is we are going to talk about those protozoa which is causing disease in human, okay. So these parasites or this protozoan parasite more precisely replicate within the host cell, okay, and based on mean of locomotion. So these parasite move, right, there are various kind of movement, okay. These parasites are categorized in four major groups, okay. The first is flagellates. Here you can see that these flagellates having a one flagella, okay, which is used for the locomotion, for movement of these parasite. Here you can see there are two very important examples are shown, that is trypanosoma and leishmania, okay. So these cause very complicated diseases, and they are basically parasite in human. If they will reach in the human, then that will cause the disease, okay. Another is amoebae. So this amoebae is like a amoeba, okay, and this they basically move from or move in the host through one specialized system which we call it as a pseudopodia, okay. They have a pseudopodia in order to make a movement, okay. And one best example, one best parasitic protozoa is the entamoeba histolytica. Here you can see this entamoeba histolytica, okay. Another is sporozoa. So these sporozoa, they don't have any flagella, they don't have a pseudopodia, so they basically glide, okay. Here you can see there are two very good example. One is Toxoplasma gondii, which again cause the disease, and Plasmodium species. There are various Plasmodium species which cause a disease in human, that is known as malaria, okay. There is a the last group of this parasitic protozoa is ciliate, okay. So these ciliates are having a numerous cilia, okay. So this numerous cilia is used for the movement, okay. And the one best example is Balantidium coli. This causes a disease, okay, but this disease is quite rare, okay. So we are not going to discuss anything about the Balantidium coli, okay. I'm just showing as a one of a ciliate protozoa parasite, okay. Another major group of parasites are here you can see, we call it as a haliments or worm, okay, worm infection, okay. So these haliments are in general they are multicellular, and please note the protozoa is microscopic. Here they are not microscopic, they are quite big parasite. They are big parasite present in the host, okay. So haliments are macro parasite, and they are multicellular, they are worm basically. They infect human but can reproduce by own, okay. There are two major phyla of these haliments. One we call it as a nematodes, okay. So nematodes are basically, here you can see that there is a one very good example, that is Ascaris limbrocoides, okay. We also call it as a roundworm. So their cross section will give the round appearance, okay. And another is flat haliments, okay, or flatworm. So this flatworm, if you make a cross section, you will see a flat kind of architect, okay. The two best example for this group of animal or parasite is Cystosoma mansoni, and another is Tinea solium, okay. We also call it as a Tinea solium as a tapeworm. It's a very long parasite, and if they infect the human, they resides in our body, and that cause a variety of serious problems, okay. So this is about the parasite. Now I will introduce about a few terms which is important in case of parasite biology, okay. So common parasitic infection, here you can see that there is a term known as case fatality rate. It's a common parasitic infection, and what is the case fatality rate? This is an epidemiologic metric that measure the portion of people. Here you can see the portion of people who died from a specific disease. Okay, let's take the example of malaria. So there are the people who died by this malaria. Okay, this is died from a specific disease among all positive cases over a period of time. Okay, this metric indicate the severity of a disease and is often used for prognosis. Okay. Here you can see that there are some diseases like leishmaniasis, sleeping sickness, echococcus infection. So they have a very high case fatality rate. Okay, this is little threatening number. Okay, compared to the other parasitic infection like schistosomiasis, guinea worm disease, malaria, okay. There is a echinococcus, leishmaniasis. So all these compared to these three diseases are quite having a risk, okay. Another term which is used is average basic reproduction number. Again, it is used in case of common parasitic infection. And what is average basic reproduction number is, it is represented as a R naught, okay, or 0. is an epidemiologic metric that describe the transmissibility, okay, of infectious agent. Okay. R0 measure the expected number of secondary infection produced by a single infectious individual in a susceptible population during mean infectious period. Okay. And here you can see that malaria has a very high average basic reproduction number. Okay. It has about 17, okay, compared to the other parasitic disease like leishmaniasis, sleeping sickness, lymphatic filariasis, so on. So now this is just a general introduction about the parasitic infection, okay, or the disease caused by parasites, or a very brief introduction. Okay. Now I will move to the to parasitic disease. First we will discuss about malaria. Okay. So you probably all are aware that this malaria is caused by Plasmodium species, which is a protozoa. Okay. And this Plasmodium are single celled eukaryotic organism. It's a single celled eukaryotic organism that belongs to the phylum Epicomplexa, okay, which is named for the apical complex. So at apical part there is a complex that is involved in host invasion. Okay. So initial symptom of malaria is quite wide range, and that includes fever. The fever is very high, and this is a periodic fever. Okay. Headache, shivering, vomiting. If not treated early, and depending on the species responsible for the disease, severe anemia can happen. I will give the detail in subsequent slide, the link how the severe anemia is happening and all those things. And I will discuss once you will learn about the life cycle of this Plasmodium species, then it is easy to understand. Here just you remember these symptoms, okay, severe anemia, metabolic acidosis, heart failure. So you will I will explain you how the heart failure can take place if there is a infection of Plasmodium. Okay. Burden on kidney or cerebral malaria may occur and even lead to death. So some of this Plasmodium infected individual, they also die by this disease. It's a quite severe. Okay. So this parasite was discovered, discovery of this malarial parasite is in gastrointestinal tract of the Anopheles, female Anopheles mosquito, led to realization that malaria was transmitted by this female Anopheles. Okay. And for this, this was done by Sir Ronald Ross, you probably are aware, and for this work he received the Nobel Prize in Physiology in 1902, okay, 1902. Okay. This is Charles Louis and Alphonse Larven. He showed the malaria is caused by a single celled organism, a protozoan of Plasmodium family, okay, which attacks the red blood cells. Okay. And he also identified other single celled parasite that cause other diseases, and for all this work he received the Nobel Prize in 1907. Okay. So this is just a simple introduction about these workers. Okay. So Plasmodium is basically, there are several species of Plasmodium. Okay. And here you can see that there are 5 human infective Plasmodium species which I am going to discuss. The first is Plasmodium falciparum. This is quite dangerous, causes the bulk of malaria associated morbidity and mortality. So, this is responsible for most of mortality is associated with Plasmodium falciparum. Okay. In sub-Saharan Africa, the Plasmodium falciparum is associated with severe malaria and complication in pregnancy. Okay. If the pregnant woman is infected with Plasmodium falciparum, it is a very complex situation. Okay. Most malaria related deaths are associated with this species, as I told you. Okay. Another is Plasmodium vivax. It's account for majority of malaria cases in Central and South America and in temperate climate, that is Southeast Asia and Ethiopia. Okay. Another is Plasmodium ovale. This is found in Africa and Asia, but especially prevalent in West Africa. Okay. Another is Plasmodium malariae, causes the mildest infection among all. Okay. Although it has been associated with splenomegaly. Splenomegaly is a situation where the size of a spleen is enlarged. Okay. Once I will explain you the life cycle, then you can understand how this splenomegaly can take place. Okay. Or renal damage. So, they also damage the kidneys, okay, upon chronic infection. Okay. The last is the Plasmodium canolosi, initially considered as a parasite of non-human primate, but cannot only cause malaria in human, but can also lead to severe and even fatal malaria complication. Okay. So, these are the 5 species which is mainly associated with malaria disease. Okay. So, this is a malaria endemic region. Here you can see that there is a near the equator, this disease is quite prominent. Okay. Now, I will discuss about the life cycle of malaria parasite. Okay. And here you can see that this malaria parasite life cycle involve 2 hosts. So, during a blood meal, a malaria infected female Anopheles mosquito inoculate the sporozoid into human host. Here you can see that the mosquito is inoculating the sporozoids in the human, the infected female Anopheles. Okay. This is inoculating the sporozoid. This is the infective form of malaria which is present in salivary gland of mosquito. Okay. So, you know that when mosquito bites, they throw the saliva in order to stop the coagulation of blood. Okay. So, this sporozoid infect. So, as soon as this sporozoid go into the human body, it will be these sporozoid immediately disappear from the blood and they infect the liver cells. Okay. And then they are over there, they these sporozoid will mature into seizoints. Okay. Here you can see there is a seizoint. This is a seizoint, and which is basically present in the liver and which ruptures and release the merozoite. Here you can see this is merozoite which is released from these liver cell. So, these merozoid basically can persist in liver, and cause relapse by invading the blood stream weeks or even year later. So, for some species, particularly the Plasmodium vivax and Plasmodium ovale, it is present in the liver. okay, in the form of hypnozoites, okay, and these hypnozoites will be remain in there. It is something like they are present in latent state, okay. But for another species, after this initial replication in liver, which we also call it as a exoerythrocytic cycle. Here you can see there is a exoerythrocytic cycle. Exoerythrocytic cycle, it means that out of the RBC. So, they are replicating in the not in RBC, okay. They are replicating in liver, okay. So, after this initial replication in liver, that is exoerythrocytic cycle, the parasite undergo asexual multiplication in erythrocyte. Here you can see that this merozoites are infecting the RBCs, and now they will initiate erythrocytic cycle. Here you can see there is a erythrocytic cycle, and then this merozoites infect RBC, and the ring stage trophozoite. Here you can see that there is a ring stage trophozoite mature into the schizont, okay, which rupture and release the merozoites, okay. Again they will release, so over there this will they will increase in number, okay, and then again they will release the schizonts, okay, and this will again infect the RBC, okay. So, some parasite differentiate into sexual erythrocytic cycle or gametes. Here you can see that there is a this bifurcate into two kind of cycle. One is asexual reproduction and another is sexual reproduction, okay, where this will form the gamete, okay, or gametocytes. So, blood stage parasites are responsible for clinical manifestation of the disease, okay. Here you can see that these gametocytes are mature and then they will make a microgametes, which is male gamete, and this is a macrogamete, which is a female gamete, and these gametes are present near the skin, blood vessels in the skin, okay. And once this again the mosquito female anopheles will, when again female anopheles will bite, then this gametocytes male and female are ingested by the anopheles. Here you can see that after bite this is again ingested by the female anopheles mosquito during blood meal. So, they take the blood meal, so these gametes will also transport it, okay. The parasite multiplication in the mosquito is known as a sporogonic cycle. Here you can see there is a sporogonic cycle, while in the mosquito stomach. So, all these things will happen in mosquito stomach. The microgamete penetrate the macrogamete and generate the zygote. Here you can see there is a generation of zygote. The zygote in turn become a motile and elongated ookinete. Here you can see this will be a ookinete, which evade the midgut wall of mosquito, where they develop into oocyst. Here you can see there is a they will penetrate and develop into the oocyst. The oocyst grow, rupture and release sporozoites. Here you can see there is a release of sporozoite, which make their way to mosquito's salivary gland. Inoculation of the sporozoite into new human host perpetuate the malaria life cycle. So, in that way this malaria life cycle is keep on going on, okay. So, after this life cycle and other information, I have a very beautiful video, and this is which is showing the life cycle of this malaria parasite in human host as well as in mosquito host, okay. Anyway, so I will give you more detail in subsequent slide. Here you can see that life cycle of malaria parasite in mosquito. I will give you a little more detail about the life cycle of malaria parasite in mosquito host. So, in mosquito's gut, here you can visualize in mosquito gut, each male gametocyte produce 8 microgametes. Okay. After 3 round of mitosis, the female gametocytes mature into macrogamete. Male microgamete are motile form with flagellate and seek the female macrogamete. The male and female gametocyte fuse forming a diploid zygote. Here you can see there is a formation of zygote, okay, which elongate into ookinete. I have explained you in previous slide also. This motile form exit from the lumen of the gut across the epithelium as an oocyst. Oocysts undergo cycle of replication and form sporozoites which move from abdomen of basically from stomach of mosquito to the salivary gland. Okay. 7 to 10 days after the mosquito feed on blood containing gametocyte, it may be armed and able to infect another human with Plasmodium species with her bite. Okay. So, this is all life cycle of this malaria parasite in human host as well as in mosquito host. Now, I will discuss about the malaria symptom. I have gave you some malaria symptom in beginning of malaria. So, here I will explain you why some symptoms are appearing. Okay. So, of course, it is causing the recurrent chill, fever and sweating. So, when these merozoites are released, okay, in RBCs, okay, there will be a release of merozoites. Okay. So, at that time there will be a peak of fever. Okay. There will be a chill and fever. Okay. And this is a kind of cyclic manner, because when the merozoites are not keep on releasing. So, they are released every about 36 to 48 hours. Okay. So, at that time the symptom will peak. Okay. So, as you can see that these parasite can infect the red blood cells or erythrocyte. So, that cause the anemia. The Plasmodium infection can cause the anemia, and there is a splenomegalia. Okay. So, splenomegalia, you can understand all blood borne antigen are moving through the spleen. So, since there is this blood is loaded with the antigen, so spleen is not able to handle. So, that is why there is an increase in size of spleen, and that result to the splenomegalia. Okay. And it is also due to the blockage of blood capillaries by merozoites. Okay. So, basically when this is happening, then that damage several vital organs. You can understand that cerebral damage, there could be a damage in brain. Okay. And this is again why, because they will block the capillaries. So, whatever the blood supplying the arteries are there, they will be blocked, right. Since it is blocked and there is no supply of blood, that may cause the cerebral damage. Okay. And due to this this blockage, that can also cause the heart failure. Okay. And you know that all blood is filtering in the kidneys. Okay. So, since the blood is loaded with this parasite, so that may also block the kidneys. Okay. So, that result to the renal failure. Okay. The symptom of malaria is not due to Plasmodium species, but it may be due to overproduction. It has been shown that there is a overproduction or kind of cytokine storm kind of thing. There is a overproduction of TNF alpha. Okay. This is why we are saying? Because the cancer patient treated with TNF alpha show malaria like symptom. Okay. So, in some cancer we treat the cancer by TNF, okay, and those patients show like a malaria symptom. That is why we can say that this is due to the overproduction of TNF alpha. So, host response to the plasmodium. So, innate immunity provide the initial defense against infection when pathogen are first encountered, when pathogen is coming to the host, which is followed by development of specific antibody secreting B lymphocytes. Okay. So, the younger children under age of 14 has a lowest immunity to malaria, and that is why there is a lot of mortality is associated with younger children. Okay. Lowest immunity to malaria, as they produce low amount of antibody against merozoite from the merozoite form of this plasmodium. Okay, you remember that there is a release of merozoite after once this sporozoite infect the liver cell, they make merozoites. Okay. It is believed that much of pathology associated with malaria is a consequence of imbalance of cytokine, okay, imbalance of cytokine or cytokine production, in which pro-inflammatory cytokine response dominate. Okay. It is believed that TLR9 nucleotide sensing receptor is involved in host immune response against plasmodium. Okay. So, there is a so this hemoglobin is converted into another form, which is we call it as a hemozoin. Okay. So, this hemozoin is sensed by the TLR9, and that may involve in production of more pro-inflammatory cytokines. Okay. So, TLR9 induces high level of TNF alpha production, leads to the clinical symptom such as fever. Okay. In addition, the membrane of infected RBC, okay, the infected RBC. So, once they infect the RBC, this RBC turn to very sticky. Okay. And this infected RBC stiffen, make a stiffness, stiffens, and it loses its deformity. Okay, contribute to the obstruction in capillaries, blood capillaries. Okay, which has life threatening consequences in severe malaria when vital organs are affected. So, once it will stiffen, then it will not go into the capillaries, and then there will be no blood supply, and if there is a no blood supply, then that will cause the cerebral damage, that may cause the heart failure, that may cause the kidney failure. Okay. So, there are drugs against plasmodium like aryl amino alcohol compound, which is basically quinine. There are various form of quinine. Here you can see quinine, chloroquine, and there are several molecules. Antifolate compound, antifols are also used in treatment of plasmodium. Artemisinin compound, you know this this work was done by Yu from China. Okay, and she received Nobel Prize in 2015. Okay, so this is quite effective against the plasmodium infection. Okay. So, chloroquine work by interfering with heme dimerization, the detoxifying biochemical process within the malaria parasite that typically yield malaria pigment hemozoin. Okay. Artemisinin was first isolated from stem, leaf, and flowers of Artemisia annua by Chinese scientist who received this. Her name is Yu in 2015. Okay. So, artemisinin effectively kill malaria parasite within host erythrocyte, which after which the dead parasite are culled by the spleen, okay, leaving formerly infected RBCs intact and circulating. Okay. So here I have a video, and you can watch the life cycle of this malaria parasite.
The malaria parasite is an ancient organism. It has been with us since before we were human. Famous victims include Alexander the Great, Genghis Khan, and George Washington. The malaria life cycle follows a devious path, swapping back and forth between mosquitoes and humans. This mosquito is infected with the malaria parasite. Because she is pregnant, she has become hungry for human blood. During the bite, she injects saliva to stop the blood from clotting. Her infected saliva also carries the malaria parasite. The parasite rides the bloodstream like a network of roads, seeking its first target, the core of your body's blood filter system, the liver. Sensing its arrival at the liver, the parasite searches for an exit. A sentinel Kupfer cell is the entry point to liver tissue. Leaving the blood, the parasite infects a liver cell, killing one or more other cells on its way. Over the next few days, the parasite undergoes hundreds of nuclear divisions, copying its DNA over and over again. A single infected liver cell can create thousands of new parasites. The next generation of parasites are modified to infect a new target: red blood cells. Inside a red blood cell, the parasite can hide from the body's immune system. The parasite slowly devours the contents of the infected cell and creates more parasites. The infected cell becomes sticky and grips onto blood vessel walls. Once mature, the infected cell bursts, spreading more parasites through the bloodstream. Malaria victims suffer fever, loss of blood, convulsions, brain damage, and coma. Countless millions have been killed by it. Most people who die from the disease are pregnant women and children under the age of 5.
So I hope you have enjoyed this video, and now you can see this the life cycle in mosquito host. Okay.
Mosquitoes are usually vegetarian, preferring to drink nectar, fruit juices, and honeydew. Only a pregnant mosquito will bite humans, seeking nutrients from blood to nourish her developing eggs. If she drinks blood from someone infected with malaria, she too becomes infected with the disease. The tiny drop of blood filling the insect's stomach is teeming with malaria parasites. The parasite form that is deadly inside humans cannot survive in the mosquito's stomach and is slowly digested with the rest of her blood meal. However, back in the human host, a few of the parasites turned into a different type of cell, one that is sexual but remains dormant. Malaria sex is triggered when the warm human blood begins to cool inside the insect's stomach. The female form of the parasite matures into an egg. The male form takes a while longer to mature into sperm. This sperm is from an earlier feed. The fertilized cell can glide and begins to explore its new environment. It migrates to the outer lining of the mosquito's stomach before transforming into a cyst. Each cyst produces thousands of thin, tiny parasites which seek out and infest the mosquito's salivary glands. The next time this mosquito bites a victim, the malaria parasite will ride in with her saliva and infect another human.
I hope you have enjoyed that video, and now you are very well clear about the life cycle of this malaria or plasmodium species in human as well as in mosquito host. So let's discuss about the malaria vaccine. There are so many, it's a huge global effort is going on in order to make a malaria vaccine. Here you can see some of the detail, and I would also like to introduce with one very interesting concept. So what people have tried, they have irradiated the mosquito, the female Anopheles, with sporozoites. Okay, they irradiated with gamma rays. Okay, and then they allowed to infect the human host. Okay, and then they were really infected with this sporozoites, and it has been shown that this is giving a very good protection. Okay. However, if you see, this is a technically very challenging. You need to make a mosquito, and then this mosquito need to be irradiated, and this mosquito need to bite the human host. Okay. So in a small setting, it may be possible, but in a population, it it is almost impossible, right? You need to make a mosquito, and then you need to irradiate, and then this mosquito should bite you. Okay, so this is little complicated. Another concept is this transmission blocking vaccine. So this is also very interesting concept in vaccine biology. Here what we do, we purify the antigen from the parasite, and from that stage of parasite which is present in mosquito, not in human. Please remember, the stage, the antigens which is present only in mosquito host. Okay, so what we can do, we can purify those antigen, and then we can challenge those antigen, or we can challenge the human by those antigen. Okay, so what will happen, then there will be a generation of antibody against that that antigen. So when the malarial parasite, when this malaria containing mosquito will bite, and then they will take the gametes. Okay, so at that time what will happen, they will take the blood as well as antibody. Okay, so this antibody will remain in the mosquito gut, and this will be effective. So they, and once this in mosquito the stage will come and that antigen will express, then these antibody can interact, and then they can neutralize those parasite. So this is the concept of transmission blocking vaccine. Okay, so there are several candidate antigen, okay, for this transmission blocking vaccine, and people are trying. So maybe in future we will have some good news, and we will have a protection against the malarial parasite. So with this I am stopping this session, and I will take one more session, and that will be the last session, and I will discuss about the African sleeping sickness. Thank you.